Hello, I'd like to share with you our study published in the British Journal of Dermatology. My name is Eric Passement. I am a senior author from a research team of Paris Cité University in France, and my team studied the genetic modifiers of neurofibromatosis 1 associated neurofibromas. The title of our study is Identification of Potential Common Genetic Modifiers of Neurofibromas, a genome-wide association study in 1,333 neurofibromatosis type 1 patients. We are a French team, which you already know because of my accent. I'm the co-PI of this study, which is carried out with co-PI Professor Pierre Wolkenstein, who is a dermatologist in Paris-Est University. And this study was part of Laurence Pacot PhD project. NF1 is a genetic disease caused by loss of function mutations in the tumor suppressor gene NF1. NF1 encodes neurofibromin, an inhibitor of the RAS-MAP kinase pathway. NF1 shows complete penetrance, pleiotropic manifestations, and variable expressivity. Only few NF1 pathogenic variants have been correlated to a specific presentation of the disease. Generally, the nature of NF1 variants has a low influence on the phenotypic variability. Intrafamilial phenotypic correlations and animal models suggested that part of the variable expressivity of NF1 could be explained by genetic modifiers independent of the NF1 locus. Specifically, previous studies have suggested the existence of modifier genes in the development of neurofibroma. In this study, we conducted a genome-wide association study to identify the genomic modifiers involved in the variable expressivity of cutaneous, subcutaneous, and plexiform neurofibromas in NF1 patients. These benign nerve sheath tumors are a major cause of morbidity in NF1 patients. We built a large NF1 cohort in collaboration with the French National Reference Center. For each NF1 patient, the phenotype was recorded and the NF1 genotype was determined. We then performed a GWAS in more than 1,300 NF1 patients for more than 7 million common variants, and we studied 3 phenotypic traits. Interestingly, we identified distinct candidate regions. A major genomic region was associated with the number of plexiform neurofibroma at the genome-wide significance threshold in the discovery cohort. Fine mapping of the 9q21 locus and functional annotation of corresponding SNPs identify candidate modifier genes using a triple mapping approach based on position, non-EQTLs, and chromatin interactions. I will just mention the example of the putative tumor suppressor Gas1 candidate gene, which is located within the same subtat than the lead SNP in neural progenitor cells. GWAS in the replication cohort identify additional candidate regions suggestive of association, as evidenced on these Miami plots for the different types of neurofibroma. Afterwards, using an original CRISPR-Cas9 based in vitro cell competition functional assay, we confirmed Gas1 and spread2 as plexiform and cutaneous neurofibroma candidate modifiers, as their inactivation had different effects on the proliferation of human immortalized NF1 mutated versus isogenic wild-type Schwann cells. In conclusion, our study highlighted several susceptibility genomic loci and identified more than 150 candidate modifier protein coding genes, with some of them involved in the RAS-MAP kinase pathway and cell cycle regulation. Distinct susceptibility loci were characterized between the different types of neurofibromas. There are several limitations to our analysis. Replication of our results from independent cohorts is warranted by possibly using a sequencing approach to detect rare variants. And it appears important that the major loci for which associations have been identified can be confirmed by functional studies, including mouse models. This GWAS should be considered as a first step in the selection of candidate genes and will fully contribute to the development of personalized care for NF1 patients. Thank you for taking the time to listen to this summary of our study, and I invite you to read our publication available in the British Journal of Dermatology. I send you my greetings from Paris.